Hi there, Mr. Mechnick with a podcast on mutations. So up to this point, we've been learning about how DNA codes for that important genetic information that helps make proteins. So we've learned the process of being able to take the DNA code in transcription to make messenger RNA, which is then transported out of the nucleus and arrives at a ribosome. And at the ribosome, we will start assembling these amino acids together into long chains called polypeptide chains, which ultimately are used to make proteins. So as you can see, this is a very complex process, and along the way, sometimes during the matching process or sometimes along the line, the DNA, when it's transcribed or when it's replicated, there could be a mistake that happens. And very often these mistakes go unnoticed, but sometimes these mistakes can be pretty significant in the organism. So we're going to focus on mutations that can alter a gene. So if you recall, there's a lot of DNA that codes for nothing. We call it the junk code. And the junk code is important because it kind of provides us a little buffering against mutations that happen. But when a mutation happens in a specific region where it codes for a gene, it can have a huge impact on that organism. So one type of mutation is called a point mutation. These occur at a single point in the DNA code. Very often we call these substitutions because one letter, GCA, in this DNA code can be altered by some factor to GTA. And as a result of this alteration, the new message now codes for the production of histidine instead of arginine. And this is pretty significant because it changes the amino acid sequence. And when you have a change like this, it's not going to stop the production of that protein. The protein is still going to be produced, but the effect is that it's going to create a lot of missense. So we've sometimes referred to a point mutation as a missense protein because you have one amino acid different and it could affect how that protein functions. One good example of this is sickle cell anemia. And in sickle cell anemia, your red blood cells that contain hemoglobin are going to be shaped differently. And the shape of that protein lacks the ability to transfer and carry oxygen like a normal hemoglobin protein does. So this is an example of missense. It will still make the amino acid. It still makes the protein, but the amino acid chain is slightly different and it can have a slight effect on the overall protein. Another substitution could happen and we could change CGU to CGA. And when this change happens, one letter is altered at one point in the DNA code. However, both CGU and CGA both code for the same amino acid, arginine. So you could have what's known as a silent mutation. So a mistake happens, but yet it still codes for the exact same amino acid. Now, very often there could be an exact piece of DNA that's removed or another piece of DNA that's added to a specific gene. So if this is your wild gene type and this is the amino acid sequence that it produces, and we end up adding an extra letter. So this is an insertion mutation where the letter G is added, and as a result it shifts everything over. So now our new codon is going to be ACA and then GAA. And GAA codes for the amino acid glutamine, which is different than lysine. So this affects every single amino acid after that point. So here you have a frame shift and it shifts everything over, which has a huge impact on that protein. It's not going to look anything like the original protein did. Sometimes you might end up with a letter deleted. So in this case, the letter A is actually removed from the sequence, and what happens is you end up shifting everything to the left in this case, and now you end up with a totally different amino acid sequence, very different than the original. So these frameshift mutations can have a huge impact. Sometimes what will happen is they'll end up creating random stop codons, so it's not even going to make the full protein. And sometimes you might have the protein have a complete malfunction. So as opposed to a missense mutation where the protein still works, a lot of times in a frameshift mutation, the mutation is causing major malfunction and the protein might not work at all. So many different types of diseases or syndromes are the result of these frame shifts. And the third type of mutation is called a chromosomal mutation. These don't usually happen during transcription or translation or during replication. This happens more so during cell division and meiosis. So when cells go through the division process and they are doing their pairing together in prophase and then separating in anaphase, sometimes what happens is there might be an entire gene removed during the division process. So you could miss an entire gene compared to the original parent chromosome. Or sometimes during that division phase, you might end up with a duplicated one. So when they separate in anaphase, it might keep copies of an extra gene present. So now you end up with an extra set of that genetic information. And sometimes during meiosis, you end up with this process called crossing over, and you can invert and put genes in different locations on chromosomes, which can have an effect on that organism as well. And the third one, or the fourth example, is what's called translocation, where genes can be basically relocated on different chromosomes in different positions. So all of these types of mutations can be pretty significant to the organism. And in people, very often during meiosis, if a chromosome is either deleted or added, you could end up with an extra or a missing chromosome. So in the case of Down syndrome, in meiosis, when this happens and an egg cell receives an extra chromosome in the 21st pair, you're going to end up having a child with Down syndrome. If a person is missing chromosome number 4, they're going to have a disease called Huntington's. Klinefelters is another example where a person has an extra X chromosome. So instead of having an X and Y for a boy, they have an X and X and another and a Y. So all of these examples are just chromosomal mutations that happen and can be pretty significant to the person that received an extra chromosome or an extra gene. However, many mutations still happen in organisms, and most mutations are neutral. They don't impact that person or that organism, so we consider them silent mutations. However, a lot of times these mutations do cause slight little variations and cause missense, and sometimes these missense mutations help an organism out. So when we think of a mutation, we can't think of some type of genetic disorder or disease, because sometimes mutations are very, very, very beneficial to the organism, and sometimes those benefits can help increase the biodiversity on this planet. Some of them, these nonsense mutations that are caused by frame shifts, can be cancer causing, and many of them, like I've said, can cause genetic disorders. So that's all I have on the podcast for mutations. So please bring any of these notes to class, any questions with you, and we will spend some more time in class going through some specific examples and doing some practice with mutations in class tomorrow.